our final two special senses are equilibrium and hearing. Now these both take place in our ear, and they both share the same receptor. The receptor in both of these senses is what's called a hair cell. So here's our fantastic image of the anatomy of the ear, and I know you'll be covering some of these anatomical structures in lab, right? But it's really important to kind of understand the anatomy as we begin to talk a bit about the physiology. So I'll run through this briefly, right? You have your outer ear, things like that auricle that kind of help funnel sound into kind of this external ear canal. Remember in the skull you had that external acoustic meatus, right? The middle ear is really going to contain the tympanic membrane, which is your eardrum. The ossicles, which are going to pass the vibrations from the eardrum on to the inner ear. Now the inner ear contains the actual structures that are going to give us our sense of equilibrium, which is balance. Right, there we're going to use these semicircular canals as well as what's called the vestibule. And for hearing, we're going to use hair cells inside of the cochlea. So those are the structures of the inner ear. Okay, so let's start out by looking at equilibrium or balanced. And balance. And really what we're looking at here is a series of structures in what's called the vestibular complex. So the vestibular complex is going to include both the semicircular ducts and this kind of enlarged area here at the base of those ducts called the vestibule. So our semicircular ducts, you'll see we have three different ducts. They're all arranged in slightly different directions. And keep in mind, all of these structures are really embedded. They're bony structures actually embedded in the temporal bone. So you had this big, if you're looking down in a skull, you had these kind of two strong ridges on either side where you'd see things like the internal acoustic meatus. These are all really like down in that bone, right? So there's this bony structure on the outside, and then there's these membranes, these fluid-filled membranes that are inside of there. So the semicircular ducts, you actually have three of them, and they're arranged in three different directions so that they can give us information. The semicircular ducts are all checking movement, or giving us information about movement. And so this gives us movement in three different planes, right? So the anterior semicircular duct, the anterior duct is going to be like if you were tilting your head up and down, like you're shaking your head yes, it's giving us information about that movement. The lateral semicircular duct is if you're shaking your head no. And then the posterior duct would be if you're kind of tilting your ear to your shoulder in the direction, right? So really kind of fascinating that we have this setup where we can learn about movement in those multiple directions. So basically what happens in the semicircular ducts, right, you have this fluid. At the base of each of these ducts, you have a region called the ampulla. So again, the semicircular at the base of each, you have this kind of enlargement called the ampulla, and within the ampulla, that's where the actual receptors, where the actual hair cells are. And these hair cells, right, this is the receptor. The hair cell, this is our most zoomed in version here, right? Notice its association here with a sensory nerve ending, right? So it's attached to a neuron. But the hair cell itself is probably called this because it has these stereocilia. They are like elongated microvilli. Yeah, check out this one, actually gets its own name because it's so big, right? But you have and I don't care if you memorize that, but you have these stereocilia that are actually embedded in this gelatinous material called the cupula. So what's going to happen here is as the fluid sloshes in these semicircular canals, when it hits the ampulla, the base, that's where the fluid is going to smash into this cupula, and then distort the hair cells. So notice here you have a whole cluster of hair cells and their associated neurons, right? And each of these hair cells, when they're showing that projection, it's actually this series of microvilli. So basically, these are mechanoreceptors. As the cupula gets pushed by the perilymph, by the fluid, endolymph, by the fluid movement, right, that's going to distort the cell membrane, and that's what's going to trigger us to go ahead and send a signal via the nerve. Okay, so that's what's going on in the semicircular canals. You also, in this vestibular complex, have an area called the vestibule. And the vestibule is this kind of bigger area between the semicircular canals and the cochlea. And in this area, you have the utricle and the saccule. Right, and these receptors are still going to be hair cells here in the vestibule, but these are going to be associated with things like gravity. So these tell you things about like the tilt of your head. So the semicircular canals were very much about movement. These are more kind of like the tilt of your head. So the semicircular canal might tell you it's going that way, but now if you just stayed here, right, it's going to be the vestibule. It also tells you about acceleration. So here in the vestibule, this utricle and saccule, basically same idea, it's giving you information in two different directions. So utricle tells you about more horizontal positioning, and the saccule is more vertical positioning. Anyway, I'm actually okay as long as you know that the vestibule is more associated kind of with gravity, the tilt, and acceleration. So semicircular ducts are movement. This is more tilt and acceleration. So when your car is pulling away from a stop sign, it's this vestibular region that's going to give you that information. Okay, so we still have the hair cells, but notice what happens here in this case, right? The hair cells and their stereocilia are still embedded in this gelatinous matrix, but now we have this really fascinating structure called an otolith. An otolith is this little crystal that sits on that gelatinous matrix. And so here they're showing you exactly what happens as you tilt the head. Those little otoliths go kind of trickling down, right? As they move, they're pushing on that gelatinous matrix, that distorts the hair cell, and this is going to give us information again. You have these little crystals in your inner ear. Kind of fascinating. Okay, so then the information, right, from those ampulla of our semicircular canals and from the vestibule, those action potentials, right, if we actually have enough change to actually cause our hair cell to trigger a neuron, all of those neurons then are going to travel down what's called the vestibular branch of the vestibulocochlear nerve. The cochlear branch is going to carry hearing, and the vestibular branch is carrying our information about movement and balance, right? So notice where this vestibular branch goes. It's this region kind of between the pons and the medulla oblongata. And notice that this information then is carried places like the cerebral cortex, right? It can enter conscious thought. That superior colliculus, that's going to be important in kind of reflexively directing eye movement. So without even realizing it, like if you're spinning, your eyes keep moving to try to find a stationary point. Anyway, that's taking place at that superior colliculi. We also send this information to the cerebellum. Remember, the cerebellum was very interested in kind of our movement patterns and comparing what we're doing to what we wanted to do. Okay, so that is equilibrium. Now let's take a minute for hearing. So hearing, then, very, very close, right, association here, right? So notice there's our semicircular canals and the vestibule. Ooh, and the vestibule actually ends up kind of being part of both. So the way that hearing, or it kind of passes through there, I guess. So the way that hearing works is our receptors, our hair cells, are back here in the cochlea. Let's see if we get a good picture here, right? So here's our cross-section of the cochlea, and we'll see that we have these membranous channels. These are going to be filled with fluid. And basically the vibrations from sound that hit your tympanic membrane are going to push fluid through these channels. And when it distorts what's called the tectorial membrane, right? So this fluid pushing in here pushes down on this membrane. That's going to interact with, you guessed it, hair cells, right? We see our same hair cells with their stereocilia, and they're going to get distorted as that tectorial membrane hits them, and we'll send a message via the vestibulocochlear nerve again. So the way this ends up working, right, is sound waves are coming through the air. They hit the tympanic membrane, and then they're basically, those vibrations are passed through what we call the auditory ossicles, right? These are the tiny bones inside of the middle ear, right? So we have the malleus. Malleus is like a mallet, right? So if this is the tympanic membrane, if this is the drum, then the mallet, right, is hitting the drum. Actually, the drum is hitting the mallet, right? But malleus, incus, and stapes looks like a little staple. At the stapes, we have this area called the oval window, where the stapes is actually able to move the fluid inside of this vestibule, which then pushes the fluids throughout the cochlea. Oh, this was really cool. They were talking about this in your book as well. If you're in an area with really loud sounds, you have this tiny little muscle that can kind of stiffen the eardrum and make it so that you're not quite as sensitive to hearing, which maybe is why when you leave a concert, like it's harder to hear quieter things, right? You're just like, my ears, but it's actually this kind of protective mechanism. Anyway, okay, so again, that pressure change in that oval window, right, from the stapes kind of pushing those vibrations, is pushing the fluid then throughout the cochlea. Right, they lay out the same thing here in figure 1730. Sound waves hitting the tympanic membrane, vibrating our three ossicles. This is a better picture maybe to see how that fluid might be moving. So you have multiple different chambers. I'm not worried about you getting into which one's the scala vestibuli and the scala tympani, but realize you're putting those vibrations into the fluid, right? And the fluid is going to be pushing on this tectorial membrane, which distorts our hair cells. Okay. The sound, the actual hearing is really kind of fascinating, right? Here they're showing you kind of how far those waves travel through the fluid, give our brain an idea of the frequency, right? So high pitch, low pitch, those sorts of things. And then kind of the amplitude of the waves is going to tell us something about how loud that is. So the hair cells get stimulated as they then send neurotransmitters to their adjacent sensory neurons. That's going to carry sound on the vestibular cochlear nerve, that cochlear branch, right? And here we see kind of the pathway where this is ending up. The inferior colliculi is part of that reflexive response to a loud sound, right? You're going to turn your head. We also see this information being passed to the thalamus our kind of sensory filter, who then passes it on to the cortex, the auditory cortex in the temporal lobe.